very nice to have all of you there. And if you like this session or any other session of Congenital Heart Academy, please share the link on YouTube to your colleagues. We really want to reach everybody with the best education possible. So our next activities, I'm going to text on the chat box, but we have the first one is going to be on Monday, is the current management and perspectives in anomalous or aortic origin of the coronary arteries. Is going to be very interesting. A lot of very skilled colleagues are going to talk to us. So it's going to be Monday on the 23rd. We are going to have a very, very nice meeting that is a meeting from our hearts. It's going to be the celebration associated with the Pediatric Cardiac Intensive Care Society. We are going to celebrate the 76th anniversary of the Blalock-Towson Thomas shunt. It's going to be a very nice, it's going to be a two-part webinars, very big, covering all of the aspects of the BT shunt. The first part is going to be on November the 3rd, and the second part, December the 7th. We really hope all of you can join us because it's going to be very interesting. And we are going to have a nice webinar on the 1st of December for cardiac critical care. We are going to talk about the interactions of what we do on the ICU and the brain and the neurological development of the patient. This is a webinar that is a joint meeting of CNOC and PCICS. We hope all of you can join us. And on the 8th of December, we are going to have a very nice session that is going to be one special session of Dr. Gil Vernovsky. He's going to have some friends with him, and he's going to talk about the cognitive dissonance in the congenital heart disease. And we are going to have a great faculty, Dr. Mary Cohen is going to talk about women and pediatric cardiology. We are going to have talks about teamwork and leadership as well. So please join us. This is going to be an amazing meeting. And talking about amazing, I want to publicly say happy birthday to Gil. Today is his birthday, and he is an amazing man and an amazing colleague. And me and Sasha, we are very glad to have him as a partner at Congenital Heart Academy. May God bless you a lot, my friend. So now we have with all of you, Dr. Anderson in another day of his amazing series.
Well, good afternoon to you all, ladies and gentlemen. You will know if you were watching last week that we completed our analysis of malformations involving the atrioventricular valves. For the remaining sessions until the end of this year, we're going to look at problems involving the outflow tracts. And of course, part of the outflow tracts are the arterial valves. So to set the scene for the sessions that we're going to have to complete this year, I'd like to discuss with you first what I mean by systemic and pulmonary outflow tracts, and suggest a means that we can analyze them. So here I'm showing you the outflow tracts from a neonatal heart. You're seeing there the infundibulum of the right ventricle. And you see that in the normal heart, at the neonatal period, the infundibulum gives rise to the pulmonary trunk. Note that I'm calling this the pulmonary trunk, much better than main pulmonary artery. And the pulmonary trunk extends to the margins of the pericardial cavity. And at the margins of the pericardial cavity, it branches into the right and left pulmonary arteries. And then in the neonatal heart, as you see here, the pulmonary trunk continues as the arterial duct. If we then turn our attention back to the heart itself, we see the aorta that is springing out from the outflow tract of the left ventricle. We can't, of course, see the outflow tract of the left ventricle, but what we can see is that as the aorta passes through its intrapericardial component, it spirals round the pulmonary trunk, leaves the pericardial cavity, And then from the arch of the aorta, it gives rise to the brachiocephalic vessels. It then continues as the isthmus, and the isthmus then joins with the arterial duct in the neonate to become the descending aorta. So what we now need to do is to separate these outflow tracts. I'm going to depend very much on what I show you today by the images that have been produced by my good friend and colleague, Shumpei Mori. And we'll come back to that when we discuss in more detail the aortic root. But what Shumpei has shown you here is the components of the left ventricular outflow tract. So there you see the intracavity part of the left ventricular outflow tract, and at the hemodynamic ventricular arterial junction, which I will be discussing with you, it gives rise to the aortic root. And then there is a part of the ascending aorta that is intrapericardial, and then the ascending aorta continues as the extrapericardial ascending aorta. So let's look at the outflow tract of the right ventricle. Now you all know, I hope, that the outflow tract of the right ventricle is dominated by the infundibulum. And the infundibulum is a freestanding muscular sleeve, and it lifts away the pulmonary root from the base of the ventricular mass. And then, as we've already discussed, from the pulmonary root, the right ventricular outflow tract continues as the pulmonary trunk, which is intrapericardial. And at the margins of the pericardium, the pulmonary trunk branches into the right and left pulmonary arteries. So I'm going to divide the outflow tracts in terms of their anatomy into the extrapericardial arteries. We'll discuss those in a subsequent session. But for today's session, we are concerned marginally with the intrapericardial arterial trunks, but we're going to be concentrating on the arterial roots. And then the arterial roots are supported by the ventricular outflow tracts. So I would like to consider the outflow tracts in terms of these four components. What I'm going to do in what remains of today's session is to discuss with you the arterial roots. And as you have already seen, the arterial roots are found in the middle of the intrapericardial parts of the outflow tracts. But before I discuss with you the anatomy of these key parts of the circulation, I'd like to share with you some knowledge about their development. Because as with the atrioventricular valves, it is now my belief that knowledge of the development permits us to understand what is going on in terms of their anatomy. And when we consider the development of the outflow tracts, we cannot ignore this key work that was published in 1942. I referred to this in the session that I devoted to development of the outflow tracts quite some time ago now, but this was the work of Theodor Kramer. And Theodor Kramer in 1942 analyzed what was going on in development. He summarized previous findings, and this, to me, is a key passage of his discussion. Because he argued at that time, a long time ago now, that it was necessary for him to redefine the terms used to describe the structures involved. And why was that? And this, to me, is the key part of his discussion, because those structures themselves rapidly change not only their shape, but also their location with ongoing temporal changes in development. And that is what I'm going to emphasize for you today. But Kramer pointed out that it was necessary to redefine how to describe the components of the outflow tract. And for better or worse, Kramer opted to use the terms truncus and conus. And you will all be well aware that Those definitions of Kramer are now very widely used when we talk about conotruncal malformations. But what I'd like to persuade you is that we now need to go one step beyond Kramer, because to me, the definitions of Kramer, truncus and conus, do not permit us to understand what is going on with the arterial roots. Because Kramer, in that stellar publication, never specified whether the arterial roots should be considered as belonging to the truncus or the conus. And indeed, if you go back and you read that work, there is still ambiguity as to what Kramer understood by the truncus. So I'd like to ask the question whether it is now better to consider development also on a tripartite fashion. As I've shown, we can do that when we consider the anatomy of the intrapericardial outflow tracts. And we now have the information that lets us look at this in far greater detail. I've already referred you to this key work that's been done in Maastricht by my very good friends and colleagues, Professor Wout Lammers, Dr. Jill Hicksburs. This work as yet is not published, but we're working on presenting a pictorial essay of the findings. Because what Jill and Wout have done is to produce interactive PDFs from the various stages of human development, from Carnegie stage 10, which is about 5 weeks of development, through to Carnegie stage 23, which is when we move into the fetal stage of development. And in these interactive PDFs, you can take apart the various components of the developing heart. So I'm showing you here the early stage when we can see the outflow tract. This is a reconstruction of the atrial chambers. There in yellow, you see myocardium of the atrioventricular canal. We discussed that a couple of weeks ago, and I showed you that in normal development, this atrioventricular canal, myocardium, becomes sequestrated as the atrial vestibules. At this early stage, the atrioventricular canal is supported exclusively by the developing left ventricle, and the left ventricle then continues within the ventricular loop as the developing right ventricle, and at this stage, the developing right ventricle is supporting the entirety of the outflow tract. Now, I also have access to the material that is present in the archive of the Human Developmental Biology resource. This is held in the University of Newcastle. It's prepared in collaboration between the University of Newcastle and Great Ormond Street, and they have an amazing series of serially sectioned human embryos. And the embryos confirm what we see here, that at the margins of the pericardial cavity, the outflow tract at this stage gives rise to the cavity of the aortic sac. So here is a section that parallels the reconstruction I've just shown you, and this is from the Human Developmental Biology resource. It's an embryo again at human embryo, Carnegie stage 13, about 5 weeks of development. And it's a section across the outflow tract. And what you can see is, as described by Kramer at this early stage, the outflow tract has a proximal part, and the proximal part bends upon itself at what we call the dogleg bend. So that then permits us to recognize a distal part of the outflow tract, and that extends to the margins of the pericardial cavity. Beyond the margins of the pericardial cavity, we find the pharyngeal mesenchyme, and percolating through the pharyngeal mesenchyme, we find the arteries of the pharyngeal arches. And they arise from the cavity within the pharyngeal mesenchyme that we call the aortic sac. Now, the key point of the developing outflow tract at this stage is that when we look at the walls, we find that the walls are exclusively myocardial. The myocardial covering of the walls at this stage goes all the way to the margins of the pericardial cavity. So that means the proximal outflow tract and the distal outflow tract at this stage have myocardial walls. And that is going to become important because what I'm going to show you is that, as Kramer pointed out, these myocardial components change their location within the outflow tract. And the myocardial border moves away from the margins of the pericardial cavity. And it is that fact that has created so many problems in producing a consensus of what is going on. So if I summarize this early stage of development, the key point, developing outflow tract has proximal and distal parts. Its cavity at the margins of the pericardial cavity becomes continuous with the cavity of the aortic sac. But here is the key point. The walls of the outflow tract are exclusively myocardial. Now here I'm moving on and I am showing you an episcopic data set. I've shown you these before. These have been prepared by my other good friend and collaborator, Dr. Tim Mohan. And Tim was initially at Mill Hill and he moved to the Crick Institute and he created these exquisite three-dimensional data sets. So this is a data set at Carnegie stage 14. And again, it's a cut through the long axis of the outflow tract. And the pinch, the waist that you see there, marks the margins of the pericardial cavity. But now there has been a major change because you see that the distal part of the outflow tract no longer has myocardial walls. That key stage has taken place. The distal myocardial border has regressed away from the margins of the pericardial cavity. And these new non-myocardial walls have grown in from the pharyngeal mesenchyme, and they are comparable to the walls of the aortic sac, but now, the key point, they are intrapericardial. There has also been growth of the dorsal wall of the aortic sac, and that dorsal wall grows in to divide the arches that become the systemic pathways, the arches that become the pulmonary pathways. So this ingrowth from the dorsal wall of the aortic sac is the aortopulmonary septum. And it is approaching the distal margin of the component of the walls that have retained their myocardial phenotype. And now these myocardial components make up an middle part and a proximal part. Those initial parts have moved away from the pericardial margins. But there is now another key point, because within these parts that have retained their myocardial walls, we have cushions. Wout prefers to call these ridges. I like to call them cushions. The key point is that these cushions, along with the aortopulmonary septum, are populated by cells that are derived from the neural crest. And we know that the influence of these neural crest cells is key to dividing the outflow tract into its aortic and pulmonary components. At this early stage, there is a foramen, an aortopulmonary foramen, between the leading edge of the aortopulmonary septum and the outflow cushions that are found within the parts of the outflow tract retaining their myocardial walls. If we move on one stage, however, and this again is another section from the Human Developmental Biology resource, it's Carnegie stage 15. It's comparable to the picture I've just shown you in the episcopic data set, but now we see, again, the distal non-myocardial part. You will note the difference in the structure, the histology, telling us this is indeed non-myocardial. And there you see the aortopulmonary septum. You can now see its continuity with the pharyngeal mesenchyme, but now it has fused with the distal margins of the major outflow cushions, and that has now separated this distal non-myocardial outflow tract into the intrapericardial arterial trunks. What we are concerned with is what happens to the middle part of the outflow tract that has retained its myocardial walls, because it is here that we see development of the arterial roots. So now, by about 6 weeks of development, non-myocardial tissues have grown into the outflow tract, and they've produced a new distal part. But we can still recognize those two initial parts, except the distal myocardial part is now the middle of the outflow tract, with the proximal part still supported by the morphologically right ventricle. So here I've gone one stage further on, Carnegie stage 16, another episcopic data set. I've cut it in the same way so that you can see the long axis of the developing outflow tracts. And now the distal component is separated into the intrapericardial aorta, the intrapericardial pulmonary trunk. But now we can concentrate on what is happening in this middle part, and we can see it transform into the arterial roots. The proximal outflow tract is still supported by the right ventricle, and as yet the cushions there have not fused. They have fused, however, in the middle part of the outflow tract. There again, you see they are not fused proximally. So now, 6 weeks of development, we can recognize a middle component that has retained its myocardial walls. And it is here we have remodeling of that cushion tissue, itself derived from the original cardiac jelly, to produce the primordiums of the arterial roots. So here is the same data set. I've just rotated the cutting plane a little bit. To concentrate there, you see the intrapericardial pulmonary trunk. And then nicely now, you see the middle part of the outflow tract. And the cushions have fused centrally. But here is the key point: two additional swellings have been developed from that cardiac jelly. And it was Kramer who first noted the presence of these swellings, and he called them intercalated valvar swellings. Now, there is some ambiguity as to how we should use intercalated, so I'm simply going to call these the valvar swellings. And I'd remind you again, the cushions have yet to fuse proximally, but they will become important. But what I've done now is I've rotated the cutting plane so that I can show you the short axis of this middle part of the developing outflow tract. And now you're looking at it from above towards the base of the ventricular mass. And there in the center of the middle part of the outflow tract, you can see how those cushions have fused. And you see that there is a condensation of the cellular tissue there. And we know from other markings that these are the neural crest cells. And it is the neural crest cells that bind together the central part of the developing outflow tract. But you see that the major cushions have not fused at their parietal margins. And those unfused parietal components interdigitate with the two swellings that are formed parietally. And there now you see the formation of the primordium from the aortic root, which is rightward and inferior, and there the pulmonary root, which is leftward and superior. But again, I should emphasize this is happening within the myocardial walls. And then as we move on, we can see how these cushions, these distal part of the cushions, will remodel to form the leaflets of the arterial valves. So now here we are at Carnegie stage 17, one stage further. You see now I'm concentrating on the aortic outflow tract. So there you see the extrapericardial aorta, the intrapericardial aorta, pulmonary root, and the aortic root. And you see already that the arterial roots are in their definitive positions. The pulmonary root, as it is formed from the outset, is superior and leftward relative to the aortic root. But at this stage, still, the aortic root is supported by the developing right ventricle. So the blood from the left ventricle has to pass through the secondary interventricular foramen. And this is where the proximal outflow cushions become important. And I'm going to show you what happens to them by changing to mouse hearts, because it's much easier to show you what is going on in the mouse heart. We have far more data sets. So this is a comparable stage in urine development to what I've shown you in human heart. There you see pulmonary outflow tract, aortic outflow tract, and the root is supported above the cavity of the right ventricle. But there you see the proximal ends of the cushions, and you see there is a notch between them because they are fusing together. And as they fuse, they build a shelf in the roof of the right ventricle so as to connect the persisting channel between the ventricles. This is the secondary interventricular foramen. The primary interventricular foramen is already remodeled to produce the inlet to the right ventricle, as we see here. So this is the same data set. I've simply rotated it so you can see it in 4-chamber plane. So you see the developing right ventricle supporting the aortic root. And there is the persisting secondary part of the interventricular foramen. The initial primary part is remodeled to surround the tricuspid part of the atrioventricular canal, which is now looking into the right ventricle. But the same data set, I've simply come a little bit anterior to show you what is happening to those cushions. The aortic root again supported by the right ventricle, but the two cushions themselves have fused together so as, like the surgeon correcting double outlet right ventricle, to create a shelf so as to tunnel the aortic root into the left ventricle through the secondary interventricular foramen. And we can see that has happened one stage further, or rather 3 stages further in human development, when the aortic root is now committed to the left ventricle. So there now you see the aortic root. The cushions themselves have muscularized and they will form the infundibulum of the right ventricle. So there now you see that the right ventricle has its 3 parts: subpulmonary infundibulum, the intrapericardial trunk, and between them, the pulmonary root. And if we take the same data set and we turn it around so that we can look in detail at the aortic root, you can now see precisely what is happening to those cushions. So there is the pulmonary root, which is now separated from the aortic root. The neural crests that were initially present between them will form what we call the conus tendon as the 2 roots separate one from the other. But there you see very beautifully that the distal margins of the cushions themselves have now remodeled to form the leaflets of the arterial valves. Additional non-myocardial tissue has grown in, and the root has expanded out of its myocardial collar, but still there is persisting myocardium in the inner heart curve. So as the aortic root is committed to the left ventricle, it retains its own infundibulum. And you see that at this stage, there is separation between the developing aortic valve, the developing mitral valve. And now if we go to the end of embryonic development, Carnegie stage 23, 8 weeks, at the stage where we begin to talk about fetal development, we can see very nicely what is happening after the aortic root has been committed to the left ventricle. So there is the aortic root. It still has a long subaortic outflow tract because at this stage, it still has a muscular infundibulum. Because there you see the tissues, the developing mitral valve, and between the two is the inner heart curvature. So eventually that is going to have to change from a myocardial to a fibrous phenotype, and we're still not sure how that comes about. But what we can see is what has happened to those proximal cushions. Because there now you see the pulmonary root, and those proximal cushions have myocardialized to form the freestanding subpulmonary infundibular sleeve. So what I've shown you is that the arterial roots form within that myocardial turret, which as they form is the middle part of the outflow tract, but initially formed the distal part of the outflow tract. And what I've shown you is that the leaflets are formed by remodeling of the distal end of those cushions. The valvar sinuses are formed by ongoing growth of that non-myocardial tissue that is initially derived from pharyngeal mesenchyme and which grows in to become non-myocardial. And then the proximal cushions themselves muscularize, form the subpulmonary infundibulum, but that inner heart curvature, which is initially part of the left ventricular outflow tract, eventually becomes converted into fibrous tissue. So myocardium is taken away in the roof of the left ventricle. So how does all that relate to the postnatal anatomy, which is going to set the scene for next week's considerations? What we can now say is that the arterial roots have significant length. And as you have seen, they are formed in the middle part of the outflow tract, and they are interposed between the ventricular outflow tracts and the intrapericardial arterial trunks. What I'm now going to emphasize is the difference in the structure of those arterial roots, because that will be key when next week we talk about malformations of the arterial roots. So let's look again at what has happened to the right ventricular outflow tract. I've now shown you how the muscularization of the proximal cushions gives us the freestanding infundibular sleeve. We've seen the roots themselves develop in the middle of the outflow tract, and we've seen that the new non-myocardial tissues growing from the pharyngeal mesenchyme have produced the intrapericardial arterial trunks. But what you now see is that in the right ventricle, because of the muscularization of the outflow cushions, the entirety of the pulmonary root has myocardial support. You can also see that there is now an extracavitary tissue area between the newly formed muscular sleeve supporting the root and the aortic root. And this was initially occupied by those neural crest cells, and part of those neural crest cells persist as the tendon of the myocardium, the tendon of the conus. The remaining tissues become this fibroadipose tissue, interposing between subpulmonary infundibulum and the aortic root. We can see that again if we turn our attention to the left ventricular outflow tract. So this is a blow-up of what you should all be familiar with, the parasternal long axis cut through the left ventricular outflow tract. And there, cranially, you see the infundibulum. You can see just a little bit of one of the leaflets of the pulmonary valve. But again, you see that extracavitary tissue area. Initially, this was made up of the cushion tissue that separated the aortic from pulmonary root, but eventually that central part becomes converted into this extracavitary fibroadipose tissue. So in the definitive heart, as you see here, there is no muscular outlet septum because the freestanding muscular infundibular sleeve separates the right ventricular outflow tract from the aortic root. And now if we look at the aortic root, we see that anterocephalad, it has myocardial support, but inferiorly, there is fibrous continuity now between the leaflets of the aortic valve and the mitral valve. Initially, that was myocardial, but for reasons we don't yet understand, the myocardium has disappeared, so that's something we need to look into, the mechanism of removal of this myocardium that initially formed the inner heart curvature. So let's now look in detail at the anatomy of the arterial roots, and we're going to start with the pulmonary root. So here I've opened out the pulmonary root, I've spread out the orifice of the pulmonary valve. So at the top you see the wall, the arterial wall of the pulmonary trunk. supported by the myocardial wall of the infundibulum. And now you see the three semilunar leaflets of the pulmonary valve. And what I want to emphasize is their semilunar hinges, because they extend distally to the sinutubular junction. Now, the sinutubular junction at the start of development marked that boundary between the myocardial and the non-myocardial components of the outflow tract. And we can see the level of the sinutubular junction when we take away the valvar leaflets. But we can also very nicely see the anatomic ventriculo-arterial junction. So this is the area over which the myocardium of the infundibulum gives way to the arterial wall of the pulmonary trunk. And it is a true ring when we open it out. You can see very nicely is that anatomic ventricular arterial junction is crossed by the semilunar hinge of the pulmonary valvar leaflets. And it is these semilunar hinges that are producing the hemodynamic ventricular arterial junction. And this has an important consequence, because it means that myocardium is incorporated at the base of all three sinuses of the pulmonary trunk, whereas there are triangles of arterial wall extending up to the level of sinutubular junction. And remember, that initially was the junction between the myocardial and the non-myocardial component of the outflow tract. So there has been ongoing regression of this myocardial border relative to what we saw during development. And that is another important point to bear in mind. But let me now build you up the perfect arterial valve as we see it in the setting of the pulmonary route. So here I'm showing you a bisected pulmonary route, and let's put in the junction between the myocardium and the arterial wall, and that is the anatomic ventriculo-arterial junction. And then let's put in the original site of the junction between the myocardial wall and the non-myocardial walls, and that is the sinutubular junction. We can now put in the leaflets. And we've seen that the leaflets reflect the location of those distal parts of the cushions that remodel. And the key feature now is the semilunar nature of their hinges. What I've emphasized is that the semilunar hinges cross the anatomic ventriculo-arterial junction. And because of that, we have triangles of arterial wall that hemodynamically are part of the ventricle. And we have crescents of myocardium that hemodynamically are part of the arterial valvar sinus, but obviously have a myocardial phenotype. Now, there is one other key area that thus far I've not mentioned at all, and that is the virtual basal ring, because it is this virtual basal ring that we construct by joining together the nadirs of attachment of these semilunar leaflets that echocardiographers call the valvar annulus. It is a ring, but it is a virtual ring. There is nothing there anatomically. And this is the key to understanding the anatomy of the arterial roots. But let's now explain again what we mean by the arterial roots. Because what I've shown you is that they extend from that virtual basal ring to the sinutubular junction. And what I've emphasized is that the moving parts are the leaflets derived from the cushions, but they are supported by the valvar sinuses. And the leaflets then are attached peripherally at the sinutubular junction, where they have zones of apposition. So let's now put all of this into context by looking at the aortic root. And here again, I come to the key images produced by my good friend and colleague, Shunpei Mori. When I first knew Shunpei, he was working in Kobe in Japan. He's now moved to the University of California in Los Angeles, and he's working there with Kalyanam Shivkumar in the Arrhythmia Center. And he's looking in detail at the wonderful images produced by McAlpine in his atlas, and he's comparing these to his own superb virtual dissections. And I'm going to be showing you today his virtual dissections that clarify for me the anatomy of the aortic root. So let's first put both roots into context. So here, Shumpei has reconstructed them from a living data set. And there you see how the aortic root is making up the centerpiece of the cardiac mass. Moved away from the base of the ventricular mass, as we have seen by the freestanding infundibulum, we have the pulmonary root. But now we're going to concentrate on the aortic root. And this is how I have usually seen it. This is one of Diane Spicer's beautiful images where she has opened the aortic root. And this is how we as anatomists have typically assessed the roots. So we can see the sinus supporting the leaflet hinged in semilunar fashion. Diane's image shows you beautifully the fibrous triangle made up of arterial wall that is extending to the level of the sinutubular junction. But she's also cut across the left coronary leaflet, and now you can see that it has height, and you can see that that height does not match its geometric height because the leaflets come together in the middle of the root. And they also come together about halfway up the length of the root. And we can see that if we look down on the aortic root from above. So the leaflets come together at the sinutubular junction, and that is what we call the commissure. But then we also need to take account of the way they fit snugly along their zones of apposition, and they extend to the centroid of the valve. And these geometric arrangements are key if we are fully to understand how the valve works. Now we can do the same thing with the aortic root as I did with the pulmonary root. We can open it out and we can cut away the leaflets. And now you see left coronary, right coronary, the non-adjacent or the non-coronary aortic valvar sinuses. And there in the triangle between the right coronary, the non-adjacent sinus, you see the membranous septum supporting the interleaflet triangle. And I show you this to emphasize that inferiorly there is fibrous support, cranially and anteriorly there is myocardium, but unlike the pulmonary root, the myocardium is incorporated only two of the valvar sinuses. We can show that particularly well in Shumpei's virtual data sets. So here we're looking at the aortic root from behind. It's wedged between the left atrium, left ventricle, infundibulum, and it extends from sinutubular junction to the virtual basal ring. But Shumpei has now reconstructed for you the anatomic ventriculo-arterial junction. And now at the level of aortic valvar sinuses, we see that myocardium incorporated in two of those sinuses. So here Shumpei has turned the ring round and has marked the sinutubular junction and the ventriculo-arterial junction with these green wires. So there you see the length of the roof. It extends from the sinutubular junction to the virtual basal ring, but now you see how in the aortic valvar sinuses, the anatomic ventriculo-arterial junction moves away from the virtual basal ring, where that myocardium is incorporated at the base of the sinuses. We can look down on the virtual basal set, on the virtual data set from above, and we can see that the sinutubular junction is circular. There, at the base of the aortic valvar sinuses, you see the myocardium, and that is differentiated from the walls by the anatomic ventriculo-arterial junction. But now look at the so-called annulus. It is not circular, it is ovoid. We can see that again if we put everything back together. Because if, as echocardiographers, you take a cut from nadir to nadir, as is often the case, you never cut the full width of the virtual basal ring. To do that, you have to go from nadir of one sinus to the zone of attachment of the leaflets on the opposite sinuses at the sinutubular junction. And this is key if we are fully to understand the anatomy of the aortic root. And Shumpei has shown that very beautifully in these series of virtual dissections he's made of living data sets. So here, now you're looking at the aortic root from above with the front of the heart downwards. So you see the two coronary sinuses, the non-coronary sinus. And again, we see that the sinutubular junction is circular. But when we look down on it in this fashion, its centroid is eccentric. But the centroid is in the middle of the ovoid virtual basal plane. And this ovoid nature of the plane reflects the way in which the aortic root is committed to the left ventricle during its development, because it has to be shifted from that initial position above the right ventricle to become interposed between the mitral valve and the septum. And we can see that again by looking at another of Shumpei's beautiful virtual data sets. There you see sinutubular junction, which is circular. We can superimpose that the mid-sinusal level, and we see there is this symmetry between sinuses, but when we put in the basal ring, it is ovoid. It is not a true circle. And if we want to measure its dimensions, we have to take note of this eccentricity. And it's only when we cut from the attachment of one leaflet to the zone of apposition of the opposing leaflets that we can properly assess what is going on. And here is just one of those cuts made by Shumpei, showing you the sinutubular junction, a little bit oblique relative to the virtual basal plane, but that permits us in this way to measure the height of the interleaflet triangle. And now we can measure the free margin of the valve. We can measure its effective height. More importantly, we can measure at the centroid the coaptation between it and its adjacent leaflets. And only when we cut the root in this fashion, from nadir of one leaflet to the opposite zone of appositions, are we able to measure the full dimensions of the root. And don't forget the interleaflet triangles. I'm going to come back to them next week when we discuss the malformations of the arterial valvar leaflets. So what I hope I've shown you is that just as last week when we were talking about the atrioventricular valvar complexes, the arterial valves are just as complex in terms of their anatomic makeup. But we can understand that on appreciating the way that the semilunar leaflets are hinged within the sinuses, but that produces a crown-like overall arrangement. Because what you echocardiographers who are watching and listening, what you call the annulus, is no more than a virtual plane that we create by joining together the nadirs of attachment of those semilunar leaflets. And it is ovoid. It is not circular. And if you want to make accurate measurements of the dimensions of the root, you have to take note of the fact that that ring is ovoid, the sinutubular junction is circular, usually the mid-sinusal level is symmetrical, but you must take note of the fact that that virtual basal ring is not an anatomic entity, and above all, it is ovoid. And that is what you need to take into account when you are making accurate measurements. And this is the basis of what we're going to discuss next week when we consider malformations of the ventricular outflow tracts.
Thanks, Bob. It was, I think, as I told you when we did the recording, was for me the first time to see such an amazing images coming from new technologies and new insight in this fantastic tool. And I think that, as you say, now it's time to make all together the idea and the anatomic images, the CT scan, and I think that Shumpei Mori is doing an amazing job in order to improve the level of assistance. And as you know, we, as a surgeon, there is a lot of interest in repair of aortic valve and a lot of technique coming up to replace also the valve with a technique, with the Japanese technique. So I think that this meeting, this presentation was really an improvement for all of us. But I see Norman Silverman directly from US. Last week you was not with us, so Norman, welcome. If you have some comment, we are waiting for you.
Yeah, Bob, that was really quite amazing. What I'm concerned about as an echocardiographer is not so much just making a dimension, but particularly for pulse Doppler, an estimate of cardiac output, which implies a cross-sectional area of the aortic valve and a velocity. And everybody has always believed that measurements of the dimension of the aorta is critical, because if you take a square of that, you square the error that you make in calculating the cardiac output. So, of course, the Doppler flow velocity is at a particular site. So where do you measure this? Do you do this in the left ventricular outflow tract, immediately underneath the valve? Is that circular? Or do you do it in the annulus of the valve, at the coaptation point? Or really, should you do that above in the ascending aorta?
Well, you need to tell me the answer to that. I mean, I've shown you what the anatomy is.
Yes.
And you're going to get fundamentally different results depending at the level at which you take it. Now, I must admit that had not occurred to me before. I mean, what for sure you've seen, and there's no question about this, is that you mentioned the annulus. First of all, you have to define what you mean by the annulus.
Right,
if you mean
right,
the
right.
virtual basal ring, then for sure the virtual basal ring is ovoid. So it would make no sense at all taking a radius there, would it? Because you can't get a full radius. And the obvious place where you get a proper ring, and I think Shumpei's images showed you that beautifully, the best perfect ring is at the level of the sinutubular junction. And the sinutubular junction is a true ring, but to the best of my knowledge, nobody has ever called that the annulus. And in many ways, that's also a paradox. So back to you. I mean, how do you think we should be doing things?
Well, I think that we have to rethink the whole issue, because I don't think anybody looks at the sinutubular junction, you know, and of course, as I mentioned, it's the annulus that wherever you take it echocardiographically, that's going to determine the cardiac output. And I think that a lot of people use the left ventricular outflow tract.
So, but what you're saying, I mean, you correct me if I'm wrong, but it's still presumed that the so-called annulus is a circle, is it not? You try to measure its radius, and you do that nadir to nadir, do you not? And
Yes.
nadir to nadir is never going to give you the full dimensions of, and particularly if it's ovoid rather than being circular.
That's correct.
So,
So I think it requires a rethinking of how to do this measurement.
I must say that the points you are now raising had not occurred to me before, because I knew you did this various things, but I've never made measurements like you do. But I mean, the point you make, there must be there must be significant error being brought in simply depending upon which measurement you take. If you're and then you say you square it, so then you're compounding the error, are you not?
That's exactly the point. Now, and the same thing perhaps is true in the pulmonary annulus as well.
Absolutely. Well, I don't think that's quite as bad, because I think that because of the freestanding infundibulum, and we've not done this for the pulmonary outflow tract, I should go back to Shumpai and I should ask him to do this, because almost certainly, and we'll discuss this next week when we talk about outflow tract malformations, and I'm going to make the point next week that because the infundibulum is a muscular tube, you have a much more circular configuration to it. So the outflow tract, the subvalvar obstructions you get in the right ventricle, as you know, are fundamentally different from the subvalvar obstructions you get in the left ventricle. That's one of the things, that's one of the reasons I emphasize so much the anatomy today and its development. So we'll discuss that next week. As I say, the potential ramifications in terms of the measurements you make, I'd not even thought of that, because Shumpei's concern was much more with how accurately you could measure the effective height and the length of coaptation. As Sasha pointed out, the Hans-Joachim Schäfers from Hamburg in Germany has been emphasizing the importance of these measurements for valvar repair. And I think that is crucially important, and that's what Shumpei has been concentrating on. We've not really considered what you're now bringing up, Norman. So, and we need to rethink that as well.
So just let me take this a little further, because it's important, particularly in the fetus, where it's so difficult to make a precise measurement, because if you make a 0.5 millimeter error, you could be making a 10% or a 15% error on a small valve. So combined ventricular output, of course, is what comes out in the fetus from both the aorta and the pulmonary artery. And to make that measurement by a Doppler measurement, you have to have good measurements of the cross-sectional area where you're measuring these things. So it becomes a very complicated issue. And I would certainly direct you to ask Shumpei to measure the pulmonary annulus and see where it's circular, so that we can learn where to make these measurements more appropriately. And I think that's the important issue here.
I mean, it's presumably, if you did 3D echo, you could begin to parallel the measurements that Shumpei has been making by doing echocardiographic measurements.
Yeah, well, that's true, except that I think I have much more reliance on accuracy of 2D for dimension measurements than I do for 3D. And yes, you're right. Of course, if you had an artificial intelligence system that could do this, machine learning, you could maybe get better measurements by 3D, but it's so difficult to do, because in order to get flow calculations, you have to measure a whole lot of things in 3D. I'm talking about 3D flow, as opposed to just a simple Doppler measurement at a particular point in time.
If we come back to the two-dimensional measurements, I mean, has anybody done comparisons if you do nadir to nadir as opposed to nadir to commissure? And have people looked?
I'm not aware of this, Bob, because everybody that I know, I mean, and I'm not saying that echocardiographers are the most brilliant people, have assumed a spherical nature to the annulus, and therefore a single dimension has been valuable. I will say that, of course, that they have preferred to do a left ventricular outflow tract and right ventricular outflow tract than above the valve. But perhaps looking at this, one has to rethink how I was doing this.
Well, and also it's a fundamental difference, as we've just been discussing, between the right ventricular outflow tract and the left ventricular outflow tract. And the interesting thing is that Shumpei sent me these exquisite images. They're from a paper he's produced quite recently, and he sent that to some clinical journals, and the clinical journals said it didn't matter. So he had to get it published in an anatomical journal.
Well, you know, that's always the case with radical free thought. When you sort of go against the grain, it's ridiculous, you know. As it appears, and there are many articles like this. I mean, if you look at Mary Ellen Avery's surfactant paper, was published not in the New England Journal, but in a journal of lesser repute, because the New England Journal turned it down because it was so radical.
Well, this is one of the advantages of the Congenital Heart Academy. We've got people all over the world that are listening to this. Hopefully they're going to be taking note. This must be an opportunity for everybody now to turn their attention to it. And we must be able to get papers from all over the world, seeing how accurately people can measure these things.
Yeah.
They'll then have difficulty getting them published, of course, but
So maybe, you know, the gold standard on this, of course, is going to be MRI. Because, but the same is true for the MRI people, is where are they going to make the cross-sectional measurements?
Well, I think, to me, CT, all Shumpei's measurements were done with computed tomography. And I think the ability to interrogate the computed tomographic data sets is just amazing. And it's, as you and I have discussed on many occasions, the ability now to look at these CT data sets, it's transforming our understandings of anatomy, not only of the normal heart, but also the congenitally malformed heart. We're going to see that as we move on.
Yeah.
No, there is.
Well, it's always sobering listening to you, Bob. I'm sorry, it's always sobering listening to you talk about the annulus, which, I mean, it's almost laughable when you see the complexity of this to make a simple cross-sectional measurement. But I think that what I should do now, I see there's a number of question and answers and chat things, is perhaps hand this back to Sasha and have somebody else who's confused make some questions to you.
No, the point is that most of the people now is very interested in this approach because of the TAVI, the new approach on the aortic annulus with the transapical percutaneous replacement of the aortic valve. And I'm sure this type of approach, Norman, is very important. As you say, probably the Academy is an opportunity to share this new view, because what we are learning from Bob, that most of the people, we have every week a lot of email coming from the people, that we are rewriting something that was only autoptic, but now with the new images coming from computed tomography and the post-processing of the tomography, we have really I remember once we met Bob here in Taormina, he told me the new pathologists can be the radiologists. It means
I used to say that about echocardiographers.
Yeah, but I don't think it works because 3D echo is not at the level at the moment.
Absolutely. No, I confirm that, and particularly for people like myself.
You know, you have Aurelio in Rome, who also works with you, and he is a fantastic computed tomographer, and he is the sort of person who is the new anatomist.
Yes. I want to tell you, Norman, probably the echo evaluation, it suffer an operator-related interpretation of the exam.
Well, you can see that from Bob's talk this morning.
Yeah.
And I think that as an echocardiographer, since I've been talking to Bob, which is not just today, but over the course of time, is that, you know, you have to think of the annulus, where you're making your measurements, as really very, very important. And perhaps the sinotubular junction is a better place to make the measurements than anywhere else on the aorta. And we'll have to see what the pulmonary CT scans show for this. I'd like to just turn it back to the audience, because there are still 180 people listening to this talk, and surely some of them have some questions that the master can answer for us.
Yes, we will ask to add the question to the question and answer section. But I have a question from Bob since I made the recording. The main message for the people: you have a lot of experience, you have seen a lot of autopsy, and you have a lot of contact with clinicians. How this new presentation, how it changed the way of how you to look to the outflow tract?
To me,
Did you change some idea? Sorry. Did you change your idea about some dogma you have then after the CT scan?
To me, it's only when I see the way that Shumpe is able to look. To me, the big problem with autopsy is that you have to open the heart to look inside it. And as soon as you open the heart, you distort what you're looking at. Now, Shumpe and I still have big problems, because Shumpe maintains that autopsy and the dissecting room is still the gold standard. I don't think that is the case. I think that now we have been superimposed by the sort of thing that Shumpe does, because he is able to take his data set and he can look at it from any plane. He can cut it any way he wants to. He can reconstruct it. He can go back to it. When I open the heart, and particularly if I cut a section across it, it's very difficult for me then to go back to that same heart and cut another section. So to me, whilst the looking at the heart is obviously important for the fine detail, that the real advantage would be to take autopsied hearts, for example, and to take a three-dimensional data set with CT, or even better with micro CT, and then do the autopsy, so that then you can go back to the three-dimensional set that you already have collected, and you can look at what you've determined from your gross dissection on the basis of the three-dimensional arrangement. Because when you have the heart in your hands and when you've opened it, you've always distorted it by making your cuts. And however well you try to put it back together, you can never put it back together in its definitive position. So I think that we have to modify what we're doing as pathologists also. And now there are, in fact, virtual autopsies. So yes, in some instances now you don't need to open the heart, because with microcomputed tomography, the data sets are so detailed that you can see what's going on. So I think everything is changing. And yes, and we need to take advantage of these new techniques.
Well, Bob, there's one further issue, and we know that in physiology, that when the aortic root receives the ejection from the left ventricle, that it itself can increase in size, and in the normal heart, this could be more, it's 17%, and in exercise, it may be even greater. So the question, even for the CT people, is where are they making their measurements? Because with this change, you need to have, if you're actually looking at things, a dynamic ability to look at the root. And we know that, for example, with cardiac output increases with exercise in young children, from my own work on my own children, for example, is that when we run them round about 100 yards and put them back on the bed to do an echocardiogram of their Doppler of their velocities, that the normal velocity, which is usually around about 1 meter per second, can go up to 2 and 3 meters per second just with exercise. So changes of velocity and dimension are critically important in determining what reality is.
I know that we have got one question, which is a very good question, Sasha, and this is from Carlos in in Venezuela who asks, does this concept influence the Ross procedure? Well, that's a very good question, because we know that the basic arrangement of the pulmonary valve, in that it has sinuses, it has leaflets, is the same as the aortic valve. But when you put the pulmonary valve into the aortic root, you cannot replicate the original arrangement of the aortic valve. So it's an excellent question, because I mean, we know that the pulmonary valve oftentimes, and correct me if I'm wrong here, Sasha, but there's increasing evidence that the pulmonary valve doesn't always behave as well as it ought to, and oftentimes it does fail when it's put into aortic position. Yes. And this could well reflect the fact that you're putting a circular infundibulum into an ovoid left ventricular outflow tract.
This is the new message. This is because I told you where you change. Carlo is a surgeon, from Venezuela. We know the way how to look this type of approach. So the question is more. We can move more when we use a bicuspid pulmonary valve, or when we have bicuspid aortic valve. How it can change the shape or the geometry of the outflow tract.
I mean, it's fascinating, in fact, that we for decades now, in fact, until I think we've based all of our assumptions on the fact, can correct me if I'm wrong, Norman, but we've based everything on the fact that the so-called annulus is a circle, and it is not a circle in the left ventricle.
Yes.
Yeah. I mean, what is the maybe Shumpei will give us an answer, because the bicuspid valve, as you're going to talk about next week, is not a simple issue.
Absolutely.
For me, as a pediatrician, looking at aortic valves, and from work from Spain as well, is that genetically the bicuspid aortic valve, when there's fusion between the left and right commissures, or when there's fusion between the right and the non-commissures, is a totally different animal, even genetically. And so it becomes very, of course, you can't use that to do a Doppler cardiac output because of the velocity changes will be unreliant, because where do you measure the velocity in that circumstance? It's not something that we do.
That's another important point you make, Norman, that hadn't really occurred to me before, that because the entrance to the root is ovoid, and so if you have right-left fusion, the parallel arrangement is going to be along the long axis of the ovoid, whereas when you have right-non, the zone of apposition then is going to be on the short axis of the ovoid. This has never occurred to me before.
Well, I think Shumpei is the guy to give us the answer, and I think the other CT people around the world, if they take up his point.
Well, we're going to discuss, I mean, you've already made the point that I'll be showing this, and indeed next week, one of the things we'll be discussing in depth is the arrangement of the bicuspid aortic valve, because at the moment, we don't even consider the difference between the sinuses and the leaflets, so we call it bicuspid. In fact, we should also be taking into account whether it's bisinuate as well as bifoliate. So to me, cusp is a terrible word. You notice I didn't use the word cusp at all today when I was describing the arterial roots, because cusp at the moment is used interchangeably by many people to describe either the sinus or the leaflet, and we have to differentiate between the two. So I think we should
in pediatrics, where I think the pathology is less advanced than adults, I think it's very rare to have a bisinuate valve.
Indeed.
I think they almost always trisinuate. And
then, as you know, oftentimes the most frequent thing you see in the infant is the unicuspid unicommissural arrangement.
Yes.
The Germans call that type 0 of the bicuspid valve, and that's a nonsense because it's not bicuspid. It's trisinuate, but unifoliate. And this is what we'll be discussing next week.
Yeah.
Well,
so Bob,
I mean, I think this is one. Sorry, Sasha. I think this is one of the great advantages of this new information that and it's propagated to a vast number of people, and I think that it makes one think about the things that one has done in terms of making measurements and accuracy and so on and so forth, and can only improve the quality of measurements that we make.
And it is fantastic that, as we say, Sasha, that through the Congenital Heart Academy, you can bring us all together and we can have these discussions, and this should be moving us forward.
Yes. So Bob, thanks we have still 150 people. I ask you, if you can invite all of them from the next week. We thanks Norman. We will see next week. And Bob, can you please close this amazing session?
Well, it's been fantastic, Sasha. I think that the discussion we've had today has been truly amazing. We're going to continue this discussion next week. So Sasha and I will be recording the presentation for next week on Tuesday, and we're going to be taking forward, and hopefully we can, what Norman has already said, we can translate our discussions today into how we approach the problems of the bicuspid valve. But we've got much more to discuss. We've got to discuss subvalvular obstruction. We're also going to consider supravalvular obstruction. And I'm going to make the point next week that, in fact, what we call supravalvular obstruction in most instances is part of the valve because it's at the sinutubular junction. So we're going to come back to the sinutubular junction.
I hope you're going to also talk about hypertrophic myopathy, which is a real big issue here, and its effect on the dynamic left ventricular outflow tract.
I shall mention it, but we've got a lot to get in next week, so I shall only give that a passing mention. But we hope we'll have even more people with us next week.
Yeah.
Sure.
Thank you.
Bye.